Hello, everybody. Welcome to Lecture 14 over Forensic Serology. One note before we get started with this virtual lecture. There will be a Forensic Serology virtual lab, posted for you later this afternoon or later this evening. You'll have an entire week to complete it, but just make sure you are on the lookout for that lab that is directly related to what we're talking about today in lecture. So these are the learning objectives for today's lecture. So you know, we'll be talking about the nature of blood. We'll talk about antibodies, blood types, Rh factors. We'll also talk about immunoassays. We'll also talk about forensics of blood and blood color tests, the origin of blood testing and individualizing blood stains. We'll very briefly talk about genes, chromosomes, alleles, hereditary, and paternity. And the reason why it's brief is because we'll have an entire chapter or lecture on that next time over forensic biology. We'll talk about the process of testing for seminal fluids, sexual assault kits, and collection of rape kits. We'll also talk about the persistence of seminal constituents in the vagina, meaning how old and how old sperm can get, and how you can tell how old they are by their degradation. Then we'll also talk about the kind of the evidence that we would collect from a rape suspect, right? Which is not necessarily the same thing as you would collect from a rape victim. All right, so first thing I want to talk about is the O.J. Simpson trial. And so if you don't know anything about the O.J. Simpson trial, this is O.J. Simpson, like famous football player. I remember this case very clearly because we were watching I can't remember if it was the 1995 or it was the 1994 NBA Finals. It was the Houston Rockets. They were either playing the Knicks or they were playing the Magic, and right in the middle of like game four or something like that, I can't remember which game it was. They cut away from the finals because O.J. Simpson had just murdered his wife and his wife's friend, Nicole Brown Simpson, and I can't remember. I think it was a Goldman or something like that. I can't remember what her wife's friend's name was, but he had just murdered her and him, and was leading the police on a chase on the California highway. And so we had to watch that for the longest time, and everybody was watching I think it was my dad and me and a bunch of like family friends were watching the finals, and very upset about the fact that the NBA finals were being interrupted by this particular chase. It wasn't even a high speed chase either. It was like a white Ford Bronco that was going like 10 miles per hour on the 405. It was the slowest chase. It wasn't even exciting. But the reason why I bring this up is because uh, if you don't already know, O.J. Simpson was actually acquitted of this case, and the reason why he was acquitted is because of a lack of standardized and well-practiced forensics. Right, the forensic collection of evidence in this case was just horrific. All kinds of contamination, all kinds of mislabeling, all kinds of stuff. It was like amateur hour. The picture you see there is pictures of the crime scene. If I actually cropped it a little bit, but if I had not cropped it and you had seen like most of it here, I know we've seen pretty bad stuff in our class already, but Nicole Brown's body was back here, uh, right around here somewhere. And so you could see it in the picture, but I cropped it because I just wanted you to see the blood evidence. But there was tons of blood evidence. Most of the evidence actually pointed to O.J. Simpson, but because of bad evidence collecting, bad cataloging, bad testing, bad chain of custody, bad contamination, all kinds of stuff, the evidence was not, viable, and so he was acquitted. The picture you actually see of him in court there is him trying on gloves, right? And the phrase that was the famous phrase at the time was, "If it doesn't fit, you must acquit." And so his attorney, Johnny Cochran, was saying that if he was the one that committed the murders and the murderer was wearing these gloves, then the gloves must fit his hands, right? And so he tried them on and it did fit his hands. But the problem was that they're leather gloves. And so if you know anything about leather gloves, they're very tight fitting. Leather gloves are super tight fitting. They don't stretch very well. And he actually had rubber gloves on, like over his hands, and then he tried to put on those leather gloves. So it was already impossible for those gloves to fit anyway. And so that was actually one of the bases for him getting his acquittal, which was completely outrageous because it was so clear that he was the one that murdered his wife, Nicole Brown Simpson. So all that being said, he even though he was acquitted of the criminal charges, he was not acquitted of the civil charges. And so, if you didn't know, there's a difference in criminal course in criminal cases. You have to have beyond reasonable doubt, right? Which is a very high bar. Anything reasonable that could be doubtful could jeopardize a case or cost a conviction or something like that. But in civil cases, all you need all you need. It's got a much lower bar, uh, and the bar is a preponderance of evidence. And so the preponderance of evidence bar was met quite easily with all the evidence, right, in the civil trial, and he ended up having to pay the family millions of dollars, and that's why he's like, he's not very well off now, right? It's an interesting case if you guys ever are interested, especially in the forensics because it was a complete cluster F, you know. Anyway, so what is the nature of blood? Blood is made up of three portions, right? Three components, three main components anyway. The plasma, which is mostly just water, right, and some salt, and all your other like uh, what we would consider not red blood cells, right? And so things like white blood cells, lymphocytes, macrocytes, your immune system, platelets, all those things are in the plasma. Um, and then uh, and then your red blood cells, which are known as erythrocytes, right? Those are things that carry the blood. Carry the blood. Sorry, they're red, of course, but they carry oxygen to and from your organs and to your lungs. Go through your blood, heart, and stuff. And then antigens, which are the bad guys, the germs and stuff, or the things that your body considers foreign from your foreign in your body, and your body will try to attack it, right, with an immune response. So how does your body attack antigens with in your blood? They do it with these things called antibodies, right? So plasma contains proteins known as antibodies, right? And they are the fundamental principle of blood typing that we use for antigen tests, right? And so um, if you didn't already know, there are four blood types, right? There's AB, and then O. And so I will talk about specifically what's what in a second, but all you need to know on this slide is that whenever you have the wrong blood type, what happens is that, say you were sick and you had to get a blood transfusion or something, and you were receiving blood and you got the wrong blood type will have an antigen in it that causes your body to reject the blood by sending out an immune response. It causes all this clumping, which you can see right here, and that's not good, right? Clumping inside your vessels will just cause like a heart attack or something, or clogged arteries, right? It's very bad, right? And so having the correct blood type is very important when you're getting a blood transfusion. So what exactly are blood types? Well, we have four blood types. It's called the ABO system because you have Group A, Group B, Group AB, and Group O, right? So when you look at a red blood cell that's in Group A, right? When you look at a real red blood cell that's in Group A, you see it has these little circly things right here, right? These little circly purple things are called the A antigen, right? And so when you have blood type A, your red blood cells have the A antigen. But in the blood itself, in the plasma, right, you have the antibody for the B antigen. What is the B antigen? Well, if you look at Group B, the B antigen is this little diamond here, right? That's on the outside of Group B, um, Group B red blood cells, right? And so since uh, Group B or blood type B has the B antigen, it has A antibodies, right? And so the thing I want you to notice here is that the A antibody has this little circular part right here, right? These little circular parts fit perfectly and attach perfectly to the antigens that are on the blood type A, right? And then also on anti B, right? The antibodies for the B, anti-B, right, which is in the blood type B, they make this triangle shape that fit this diamond perfectly on the antigen of B. So if a person was group A or blood type A and they accidentally received group B blood or blood type B, right, it'd be catastrophic for them. And the reason why is because the antibodies that have the anti-B, right, will start attaching themselves onto the blood type B antigens, right, and start clumping up the blood, which uh, can cause all kinds of blood clots, right, and blood clots are very bad. So if you're group B, sorry, if you're group AB, right, or your blood type AB, uh, you actually have both antigens. You have the B and the A antigen, right? The circle and the diamond. And your blood actually has no antigens, no antibodies in it at all, which is great because what this makes you is called the universal recipient. And being a universal recipient means that you can actually you can actually receive blood from any other blood type and you won't have any problems because you don't have any antibodies, right? B or A. And so you can receive B blood, A blood, O blood, and nothing will happen because there are no antibodies there to to clump up your blood. That makes you the universal recipient. You can receive blood from anybody. The last group is O type or blood type O, right? Or O blood type, and I'm actually O negative, right? So what that is that you actually have no antigens at all on your red blood cells. That's me right there, right? And but the problem is that I have the antibodies though for both A and B antigens, right? That's not good because if I accidentally get blood from A, B, or AB with all those antigens present, my antibodies are just going to attack like crazy these antigens that are floating around, right? Clumping up and then giving me blood clots, which is a bad thing. So, but the good thing about being O though is that it makes me the people like me who have O blood type the universal recipient. The universal donor, which means that I can actually donate my blood to anybody of any blood type and they'll be fine since my blood doesn't have any antigens in it, right? So that's called being O type, right? So the most of the population actually is blood type O, right? 43% of the population is blood type O. 42 is blood type A. Then we have coming in third, blood type B with 12%. And a very few people are blood type AB, where they're the universal recipients. Only 3% are universal recipients. Those guys are lucky, right? But I feel lucky too that I can actually donate blood to anybody, right, as being an O. There's also another blood type, and that blood type is the RH system, and we'll talk about the next, right? Has to do with actually the RH factor, right? And the RH factor has to do with uh, 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 this thing called the D antigen, right? So you know how over here we had the A antigen, the B antigen, right? But now we have this another antigen called the D antigen, and it's most, I guess you would say it's most uh, worrisome, concerning when it comes to getting pregnant, right? And so if you look at a person who is Rh positive, right, or D antigen positive, you can look at their red blood cells, and they have these like spikes on them, right? But if they're Rh negative, then they're smooth. They don't have the D antigen on them at all. So why does that? Why does having either Rh positive or Rh negative have anything to do, any kind of like adverse side effects when it comes to getting pregnant or having a baby? Well, say you have a man who's Rh positive, right? Which means all his blood cells have this weird spike on them, the D antigen. Okay. The first time that he and his wife or whoever get pregnant, right? Everything's fine. Okay, everything's fine because, uh, well, sorry. Let me first say the woman is, let's say the woman is Rh negative though, right? So she doesn't have any spikes or any D antigens on her red blood cells. Now, the first time the man and the woman uh, uh, get pregnant, right, the baby will be Rh positive because of the man, right? But the woman will be Rh negative because she was always Rh negative. And for the first pregnancy, right, for during the first pregnancy, um, everything's fine, right? And the first baby that she gives birth to will be fine. There will be no complications, right? But the problem is that when she delivers the baby, there's a lot of blood happening, a lot of blood mixing and fluids mixing between the fetus and the mother. And so some of the kids, the babies or the fetuses, Rh positive blood will enter the mom, which is perfectly fine, right? Except that the mom will produce antibodies for the Rh positive blood, right? Or the blood with a D antigen or the spikes on it, okay? And essentially get rid of all of those foreign blood cells, right? Which sounds like a great thing, but if she ever decides to get pregnant again by that same person, by the same male, right, same man, which is likely, right, the baby will again be Rh positive, right? But the problem now is that the woman has Rh antigen, Rh antibodies, right, that will attack the baby, essentially aborting the fetus, right? And so the that's really the problem right there, right? The antibodies will literally attack the fetal red blood cells, right? And often the fetus doesn't survive. And so the problem there is that the man's always the problem, right? Because he's the one that's Rh positive, and he's the one that made the baby Rh positive to begin with, which caused the woman to make Rh antibodies, which will attack a second baby, right? So again, the first baby will be fine, but when the baby is given, when the mother gives birth or during delivery, there's a lot of mixing between uh, there's a lot of mixing between the fetal the fetal body fluids and the mother's body fluids, and so all these Rh factor blood cells will enter the mom, and the mom will make antibodies to fight those Rh factor blood cells because it's foreign. But if the woman gets pregnant again by the same man, which is likely, again, the fetal blood cells will be attacked by its own by the mother's antibodies, which is not good for the fetus, right? So that's why the fifth uh, that's why the other antigen, right, or the other blood typing is also very important too. It's important when having getting pregnant, and you should always, if you're going to, you know, if you're going to um, have a baby and get pregnant, it's really important to make sure that the male is whether they are Rh positive or Rh negative, because if it's caught in time, there's medications for it, right? Or you can, make sure that the second baby is safe and all that stuff. So just making sure, it's important to make sure that the male is if the male is Rh negative, then no big deal, right? But if the male is Rh positive, you definitely need to check that out. So here's your attendance question for actually watching this video today. Which of the following blood types contains only A antibodies? I'll give you a second to answer the question. And if you don't know the answer, it's in the previous slide. Okay? Is it A, B, AB, or O? Do 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 do. Do 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 do. Do, 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 do. All right, go ahead and answer the question. Don't forget to use your QR code for attendance, right? Making sure you're actually watching this video. All right. So one of the most common things that we use to one of the most common things that we use to test for in forensics and forensic serology is called the immunoassay, right? And so it's again it's an antigen antibody based assay. And so what we can do here is, in this particular immunoassay, we can actually create antibodies, right, that will detect drugs. And so, say you have a drug molecule, any kind of drug molecule, and what you do is you attach a protein carrier to it, right? And when you attach a protein carrier to it, you can inject it into a rabbit, and the rabbit will make protein antibodies, right? And then you can use those protein antibodies to detect the presence of drugs, right? Uh, uh, in anywhere, right? In blood, in urine, uh, all kinds of body fluids that are from humans. So amino acids are pretty common. I keep on saying amino acids. I'm sorry. Amino assays are pretty common in forensics. So how do you actually make an antibody, right? There's a couple of different antibodies first to note of. There's the polyclonal antibody, right? And these antibodies are ones that come from several sources, right? Whereas uh, there is a one called a monoclonal antibody, right? And that comes from a single source, right? A single source. And so the way that we make antibodies is by injecting an antigen into a small rodent, right, a mouse, and the antigen is like again anything foreign, and so the rat will or the mouse will go through rabbit. Oh, how's that? It could be a rabbit too, right? Will go through and begin to create antibodies for the foreign antigen that you have introduced to its blood, right? And once that happens, right, what we can do is you can remove the spleen from the rabbit or the mouse, right, and isolate the cells so that we can get the antibodies, right, that are made because of the antigen that we injected, right? And so uh, this particular thing here, what's happening here is we can have a malignant blood cell, right, attached to the spleen cell, right? And then all this stuff happens that you don't really need to worry about, right? But what happens is that we have a whole bunch of spleen cells get grown, right? And the spleen cells make a whole bunch of antibodies, right? This part right here is kind of beyond the scope of the class, right? But just know that we can go from a few spleen cells to a whole bunch of spleen cells, right? And from that we can get a whole bunch of monoclonal antibodies, right? To detect, for example, that drug molecule. So how do we detect the drug molecule, right? The specific assay is known as the EMIT, right? Or the enzyme multiplied immunoassay technique. And how does it work, right? Well, the first thing that we do, right, is that we have our drug antibody, right here, that's on like a surface, okay? And that remember that antibody comes from like the mouse spleen cells. It's kind of sad that we have to take its spleen, but yeah. And then we'll actually flood the the antibodies with a labeled drug, right? A drug molecule that we've labeled so that we know that this is our standard, right? A labeled drug molecule. And so if there's no drugs present in the urine, right, all of our antibodies will have labeled drugs on them, right? They'll all have this little asterisk that we know is what we provided. But if there is any drug present in the urine, right? So just regular drugs, not the drugs labeled by us so we know it came from us, right? Then they compete for the antibody sites, right? And so what you'll see is half of the antibody sites get labeled with drugs that are labeled, and the other half will be labeled with unlabeled drugs, or will be attached with unlabeled drugs. And those unlabeled drugs are drugs that were present in the urine because the suspect or whoever was getting drug tested uh, actually had like illegal drugs in their system, right? So that's how you do the emit assay. Um, I think the more common thing that people see though is using the forensic serology to do blood testing, right? And so one of the things that we use to do blood testing or to see if there's human blood present, uh, you probably heard of this already, but it's called luminol, right? And so whenever you're at a scene and there's a whole bunch of like droplets everywhere, or maybe there's not any, you think there was supposed to be blood drops somewhere, blood stain patterns all over the place, right? You can spray this liquid on there They're called luminol, and then when you turn off the lights, the blood will react with the luminol, making a glow purpley colored, right? Which is pretty cool. But it's gotta be super dark when you do it, and it's not very bright, and so it's hard to take pictures of. And so they've actually moved on to a different one now, and the one that you see down here is actually called leucomalachite green. And so in the presence of blood, leucomalachite green will actually turn this like very bright blue color. It's very pretty, right? So why do we even have this test called glucomalachite green to see if blood is actually blood? Well, the first thing is, again, the question would be, is it actually blood? It could be ketchup for you all you know, and ketchup will turn glucomalachite green that color, right? The next kind of question you have to ask yourself is the species blood is what is the species that the blood came from, right? It might not even been human blood. It could have been chicken blood, right? Pig blood. It could have been anything. So we always have to test it to see if it's human blood, right? And if the blood is human, right, how closely can we associate it with the individual, right? Can we actually attach that blood with an individual, individualize that blood with a person, right, a suspect or a victim? So there's ways to do that, but the most common way now, what we do now is with DNA, right? So there's a couple of different other kinds of like drug tests that you can do to see if blood is blood, right? If blood is present, because oftentimes when you get to a crime scene, blood's been washed away by bleach or something like that. So one good one is called the Kastle-Meyer test, right? The Kastle-Meyer test reacts with hemoglobin. It turns the blood a deep pink color, right? There's also the Blue Star test, right? The picture you see here is the Blue Star test right here. You see the sink here is empty. The guy's hands here look kind of clean, right? You spray it with the Blue Star chemical and you turn off the lights, it turns bright blue. Same thing with his arms, right? So the Blue Star test is pretty cool. Um, and then luminol, right? Luminol is there too, right? I already talked about luminol a little bit. Uh, Luminol is actually the good thing about luminol is that it's very sensitive. Like it can detect very tiny amounts of blood, but again, the problem is that it's pretty dark. You got to get it really dark to see it, right? But yeah, so that's really helpful when someone has gone through and try to clean up the mess and stuff. Luminol is really helpful. And again, the one I did mention a second ago is not listed here, but the one that we really use the most now, like especially at the Dallas County Crime Lab, is the leukomalachite green test, right? Which is the picture I showed you there. So, excuse me. How do we know then blood is actually human blood and not like somebody else's, some other animal's blood, right? What we can do is we can actually mix the blood, human blood, right? What we think is human blood with rabbit serum, which is just like rabbit plasma, everything but the red blood cells. And so when you do that, this is called the precipitin test because there's this little layer here, right, of precipitin that forms between uh, the human blood and the rabbit serum, right? And so if that forms, then that might possibly be human blood, right? So once a stain has been characterized as blood, a precipitin test will determine whether the stain is of human or animal origin. And so if it's human origin, we see this little precipitin thing, this tiny uh, little band that forms between the blood and the human blood and the rabbit blood or the rabbit serum. There's also another one called so you can also do a precipitin test using gel effusion. Sorry, gel diffusion. My apologies, gel diffusion, and this one's actually a little bit easier. You can add your uh, let's see here. What are we doing? Antigen antibody are added to their respective wells, and antigen and antibody move towards each other. Antigen and antibody have formed precipitin between the gel lengths. Okay, so the extracted blood stain and the human antiserum are placed in separate holes, right, on each of the gel, right? So yeah, right there and right there. If the heat blood is human, a precipitin line will form where antigens and antibodies meet. So here's our line right here, right? And if that meets, then you know it's human blood. I've never actually done that test before. I've seen this one, but I've never done this test before. They're always coming up with these like new little cards and kits to make your forensic biology testing faster and easier, right? And so again, I've told you this already before, but the way that we individualize blood stains now, or we identify where a blood stain came from is using DNA. And so DNA is well, it's not the probably, it is. It's the gold standard right now for identifying all kinds of biological fluids. So we'll have a long chapter or a long lecture just based on forensic biology, which is DNA, right? But just a quick thing about DNA though, and the genes and chromosomes, right? The gene is the basic unit of heredity, right? So when you get things about yourself from your parents, your mom and your dad, characteristics about yourself, like your eye color, your hair color, your skin color, all that kind of stuff, you inherit it through a gene, right? This is the basic unit of heredity, right? Which is the gene, right? So you get the blue eye gene from your parents. That's the gene, right? And so human DNA is actually wrapped up in these things called chromosomes, right? They look like little X's, except for the Y chromosome. It looks like a Y, which you saw that under a microscope already. Those are chromosomes. And so excuse me, each gene is composed of DNA, and it's designed to carry information, right, to identify what a purpose of a cell is, or the job of anything in your body, right? Characteristics of you in your body, right? And so, the DNA is in the gene, right? Which is just the entire chromosome is made of DNA, right? But part of the DNA, right, is got a gene in it, and it gets wrapped up into these things called chromosomes that are in the shape of X's and Y's, which you've seen before, right? Humans have about 30,000 genes, right, that we've identified. And so uh, every location on a chromosome that a gene is located, because not all of your chromosomes actually have genes located on them, but every location on a chromosome uh, that's filled with DNA that has an active gene uh, that that's called a locus, which is just another fun word, I guess, for location of the gene. I just call it location, right? So just some more quick facts about chromosomes. Humans contain 46 chromosomes. And the reason why is because we get 23 from our mom and 23 from our dad, right? And so during fertilization, a sperm and egg combine, right, and each contribute 23 chromosomes. So that's why each person has 46, right? Once the chromosomes form a new cell or a zygote, right, that those 46 chromosomes will work together to develop your offspring, right? So at each loci, right, which is just the plural of locus, right, for a gene, you have an allele. And alleles are just different versions of the same gene. Okay. And so usually, usually there's two alleles, right, present. And if you have one allele that's different from the other allele, then we call that a heterozygous gene pair, right, or two different alleles. If you have a homozygous, that means you have two identical alleles, right? And so when you have a heterozygous, one of them is usually dominant while the other one is recessive, right? So if we took a look at this example down here, right? We can look at these different gene loci. Here we have gene P. I have no idea what gene P does, but gene P here are both two big P's, right? So both of those are the dominant P's, right? No little P's. We would call this homozygous. Specifically, it's homozygous dominant, right? Because they're two big P's. Here at another gene, we have two little a's, right? Both alleles, again, these alleles always come in pairs. Both of these are little a's right here. So again, this is homozygous, right? And since they're two small a's, we call them recessive. So we call that homozygous recessive. And then this is probably the most interesting one, which is where we have a big B and a little b, right? The big one is the dominant allele, right? The big B. The little b is the recessive allele. And so we call this heterozygous, right? And so when you're heterozygous, the big B's traits are what will show up as a characteristic in a person, right? And so I think that the easiest way to understand this is maybe, and this is a very simplified example, but let's use hair color as an example, right? So generally, red hair color is actually recessive, right? And so a kid that has, or a person who has red hair color, since it's a recessive gene, uh, it's the red hair color gene. Both alleles are probably recessive, right? Where they're both little letters, like small letters, right? Not big letters, not capitalized letters. Okay, and so it's very unusual then to not unusual, but if you were to have a heterozygous, right, and say the red-haired kid marries or red-haired person marries a dark-haired person, right? Dark hair is actually dominant, right? And so say they come together and they have a kid, right? Their kid will most likely be heterozygous. One with a small b coming from the red-haired uh, partner, and the other one uh, having a big B from the dark-haired partner, right? And so their kid will end up being heterozygous, one recessive and one dominant. But the dominant allele, right, will be the one that shows itself as manifests itself as a characteristic in the child. And so most commonly, what will happen is that the child will have dark hair and not have red hair, right? But there is a chance that the child could have red hair too. So using these alleles, we can actually determine heredity and paternity, right? And so if you take a look at the graphic I have right here, right? If you look at the first image of this guy right here, right? This male contributor, right? You see that he has alleles present here, 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 and here, right? A black one, two gray ones, a thin gray one, another gray, and then just a thin black one, right? And the mother right here has three fat gray ones, right, and a thin gray one down there, right? And so when they come together and they have a child, right, remember that the male will donate its DNA to the child, and then the female will also donate its DNA to the child in the form of 23 chromosomes per person, right, for a total of 46 chromosomes. They mix to each other. And so, you see here now that the child has the gray one from the mom, the black one from the dad, that gray one from the dad, that gray one from the mom, those two gray one, that gray one from the dad, that gray one from the mom, the light gray here from the dad, dark gray from the dad, black from the dad, and then the gray from the mom, right? So the baby, right, the offspring, the child, will have a combination of both parents' alleles. Okay, so this is a really good way of doing a paternity test because if you look at a child, right, and look at their DNA profile, right, you can see here the mother has matching DNA profiles with the child, right? So that's definitely the mother there because the child and the mother both have this fat gray hair here, the fat gray right there, fat gray there, and the light gray or the thin gray one. But if you're checking the father to see if this father here is the father, right, or this male here is the father, right, you can see that the child does not have a matching thin black allele right there, or a fat black allele right there, fat gray allele there, or even a fat gray allele there, or even a fat gray slash a fat gray allele there, right? In fact, the only thing that matches is this allele here, this thin black one. So since not many of the alleles match with the child, that means that this male here did not contribute to the child's DNA. Therefore, he can't be the father. So if you were watching Maury Povich, you'd be like, you are not the father of this baby, right? So that's how we can use alleles and paternity uh, and heredity to figure out the father of a child, right? We'll go through this in a lot more detail in forensic biology, but this is just like kind of like a very simplified example. And so now we go into talking about testing seminal stains, right? So oftentimes you will find biological samples and you don't know that it's actually sperm stains, right? A seminal stain or a sperm stain. which is important for a sexual assault case. And so, one of the best things, one of the ways that we can characterize uh, the stain, what we think a biological stain is actually sperm or seminal fluid, is using this thing called the acid phosphatase test, right? And that's a color test. This color test will turn purple, right, in the presence of the acid phosphatase enzyme, which is an enzyme that is an enzyme or a molecule that is present in seminal fluid, right? So this is basically the picture right here, right? It turns purple in the presence of seminal fluid. The other way we can also do it too is also by looking at it under a microscope. You'll be looking at some sperm under the microscope in lab today. When I post the virtual lab for you. Excuse me. When I post a virtual lab for you, and so you can also tell it's sperm because sperm can, and that's actually probably the most common way people do it in the forensics lab. Um, they'll maybe do it in the field using the acid phosphate test, but in the forensic lab, they'll just look at the sperm under the microscope to see if it has the head and the tail, right? Other testing you can do too, the P30 test, right? And so once you have confirmed that this is sperm, there's sperm present in this fluid, in this biological fluid, then you can go ahead and do DNA typing on it and figure out whether or not the person, uh, figure out the DNA profile from the sperm sample. So what are the kinds of evidence that we collect from a sexual assault victim? All right, and so this is usually done at a hospital with a forensic nurse. And so it's important that whenever a female or even a male has been sexually assaulted, that they don't go take a shower, which is probably the most common thing that people do. They're washing away all the evidence, which is really bad. And so if something happens, it's best to get straight to the hospital, report the sexual assault, and have a rape kit done, right? Or sexual assault kit done. And so at Dallas County, when in the mornings, we would always go over to Parkland Hospital because rape kits were done there, and we'd pick up so many rape kits from the hospital and bring them back to the lab that were being done from overnight every day. We would, right? So, and you'll see the rape kit that we actually use in a second. So you have to do pubic combings, right, in a rape kit. That's one of the kinds of evidence that's collected because the perpetrator, while performing the assault, could leave behind their own pubic hairs on the victim's pubic hairs, right? Then you also have to extract pubic hair controls from the victim. Usually they'll extract 25 or so pubic hairs from the victim just to use as elimination of any like loose pubic combings that might not be from the suspect. There's also always swabs, external genital swabs done to see if there's any kind of seminal fluid or sperm or anything that was left over after the assault. That's still on the external uh, genitalia of whoever, whether it's a male or female, right? So that includes then like vaginal swabs, right? I mean, they also do vaginal swabs too, in case there was any seminal fluid left behind, sperm left behind in the semen, right? Cervical swabs as well. Again, same reason. And also rectal swabs for the same reason, because seminal fluid can be left behind in the rectum as well. I think one of the things that people don't commonly think of when we do a sexual assault case is that we also collect swabs of body areas such as the breast. And the reason why is because during a sexual assault, oftentimes the perpetrator is kissing or licking the victim on all kinds of body parts, not just the breast, but also their arms, their legs, their feet, their face, their neck, all kinds of places. So you have to be swabbed all over your body just in case um, licked or kissed the victim there, right? Of course, there's oral swabs too, in case during the sexual assault there was any sperm or semen deposited uh, in the victim's mouth. Also, head hairs. We'll also take a blood sample. We'll also do a buccal swab. The buccal swab serves two purposes. It does It collects, again, if there's any seminal fluid or sperm present in the victim's mouth, but also we use a buccal swab for collecting the victim's DNA, right, for doing, again, elimination purposes, right? Fingernail scrapings are always done too to see because oftentimes there's a struggle and the female scratches the suspect, right, and then their the suspect's cells, skin cells, epithelial cells will end up under the fingernails of the victim. A urine sample and then all the clothing, because sometimes whenever a sexual assault occurs, the victim may not be completely declothed; they might be still have some clothing on them, and then while during the sexual assault, the um, the perpetrator could ejaculate and leave behind semen or seminal fluid on the clothing of the victim, right? So all outer and undergarments should be carefully removed and packaged separately in paper. Remember, whenever we have biological evidence, we always package it in paper because if you put it in plastic, it'll just mold, and mold will kill the DNA, destroy the DNA. So no plastic bags, always paper. Remember that for your crime scene. And then if the sexual assault occurred on a bed, all the bedding is collected, right? And then, or any object that the assault took place. And usually what happens in Dallas County is we'll take the entire bedding back and examine it in the lab itself. We have these big tables, these huge tables, and we'll put the entire comforter on the table or something. And we'll have the blue crime light, which you have to use in the crime scene, the first crime scene, and we'll shine it all over the comforter to see if we can find any seminal fluid, right, or sperm samples on the bedding or the comforter, which is not an easy job, by the way. So this is what a rape kit looks like, right? These are all the components of rape kit. Everything goes into this box, though, right here. And so when we go and collect those from the hospital, Parkland Hospital, when I was at Dallas County, they always came in these boxes. So we had a whole bunch of these boxes. We would take them back to the lab and we do this thing called triage, which Marlo taught you what triage is. That you go through and kind of just catalog all the evidence. And again, what's in this rape kit, right? Uh, we have evidence collection bags for foreign material, for outer clothing, for underpants, debris, pubic hair combings, pulled pubic hair, which are the pubic hair standards, right? Or what do I call them over here? Controls, vaginal swabs, rectal swabs. We also have swab boxes and microscope slides for looking at the sperm under the microscope, oral swabs and smears, saliva samples, blood samples, and also anatomical drawings from the victim showing where they were assaulted. And then, so this is just a particular a slide that kind of gives you a timeline on how sperm survives in the vagina, right? So if you see sperm with tails, right, that means that the sperm has been in the vagina for about 4 to 6 hours, right? If you see sperm without any tails, then it's been there for 3 to 6 days, right? You can do the acid phosphatase test, right? And if it's positive, that means the assault occurred less than 48 hours ago. And when you do the prostate specific antigen test, right? That means that it happened less than 72 hours ago, right? So you can kind of really narrow down it within a day or so of the assault, right? Like especially with this part right here, right? And then with this part right here, right? So you see sperm without any tails at all in a swab, right? That means it's been 3 to 6 days since the assault happened, right? After 6 days, you don't even see the heads anymore. But for 4 to 6 hours to 48 to 72 hours, you can really narrow down the time frame. So we also collect evidence from the suspect as well. And so, sorry, I didn't mean to move my laptop there, but we also collected from the suspect as well. And so, uh, hopefully the suspect has been apprehended within 4 hours of the assault because, again, just like the victim, the suspect could wash their hands or take a shower and we'd lose all our evidence, right? because we really want to get to see if we have any of the victim's DNA on the suspect's underwear, right, or even on a penile swab, right? See if there's any of the victim's DNA on on a penile swab, a swab of the suspect's penis, or the vaginal swab, right, of the suspect, because females can also rape or sexually assault males, right? We also collect from the suspect all the clothing that they were wearing during the assault and a sampling of their pubic hair, right, and their head hair. Again, penile swabs or vaginal swabs, depending on the sex of the perpetrator, the suspect, right. Blood samples, buccal swab for DNA typing, and then the forceful physical contact between the victim and the assailant may result in the transfer of physical evidence such as skin cells. So, like I said, the epithelial cells that will end up under the fingernails, so of the victim, or the victim's skin cells will be uh, on the suspect through a struggle, right? Maybe the victim scratched the suspect, and so not only did the victim have the suspect's DNA epithelial cells under their fingernail, but the victim left behind their own DNA on the suspect in the scratching process, right? Of course, blood, semen, saliva, hairs, and all those things can be on the victim. I mean, on the suspect that we need to collect. So this picture that you see here, you should actually already recognize, but I'll tell you what it is. These are actually cheek cells or from a buccal swab, right? And so this is like the most common way of collecting a control sample of DNA from either a suspect or a victim. We do a cheek swab or a buccal swab. Some people call it a buccal swab, but I call it a buccal swab. And so, actually, whenever you join a law enforcement agency or forensic science lab or whatever, or a crime lab, they make you do a buccal swab so they can get your DNA, just so they can have your DNA profile on file for elimination purposes because you might leave behind your DNA uh, at a crime scene by accident. All right, so these are the boxes that we pick up from the hospital every morning from all the rape kits that were done overnight. All right, and so before collecting biological evidence, it must be photographed as always. Nothing happens unless it's been photographed, and also recorded in sketches, thoroughly documented in handwritten notes. Right, always wear latex gloves when collecting the evidence for a like latex or nitrile. If you're allergic to not latex, just use nitrile gloves. Right, to collect the evidence. Don't forget, don't use anything plastic or airtight containers because mold will grow, which is bad, right? So always use paper. Put it all into a well ventilated box, like these boxes right here, which we get at crimes at the Dallas County Crime Lab. And then once you get it and you've triaged it, you can put it into a store. You can store it in a refrigerator or cold storage location uh, until time for analysis. And then that's basically it. That's our lecture for today. Just remember that there will be a forensic serology lab posted for you later this evening, if not tomorrow morning, or it might even actually be tomorrow morning. But you'll have an entire week to do it from home. And I will see you in class on Thursday. Thank you.